one, right? It's on the outside. Yep, exactly. Inside, the vitreous humor, it's a little tricky. Don't worry about this image because I'm gonna talk about vitreous humor when we get to our image. The retina is actually this beige, very, very thin tissue. Okay, and that's actually the optic nerve. On the other side, it comes out there. And so, oh, I think it's the optic nerve. Wait, did I lie? It doesn't actually matter because we need to know the optic nerve from the outside, not the inside. The optic disc will be on the inside. So basically, from this image, though, you can see that we've got the retina, which is the very beige-ish tissue. The black part is the choroid, and of course, the white part is the sclera. Don't worry about all the tissue on the outside. Okay, that's really it. It's actually not a big deal. We don't have to do the lens. So let's just now. This is a really good image of the vitreous humor. That is a good image. It's not like that's what it is. And also, this is a really good image of the ciliary body, even though it looks kind of inverted the way the model is, like with the color. But this is where the pupil would be here. The iris is just around there. And then you've got the ciliary body, right, like that. So this is actually a really nice image, kind of highlighting that. Again, the black part is the choroid, and the white part, of course, is the sclera. And in this image, you can't actually see the retina because it's the front of the eye. All right, awesome. So again, not part of ours, but you can see the cornea here. And then the sclera is just the white part. We don't care about this because it's not. Now, this is a good image, another good image that's not ours, of the vitreous humor. You can see this gelatinous substance there. And then here, beautiful, very, very thin tissue-like retina. The black part is the choroid, and again, sclera here. Now, the cornea in this image is going to be right here on the very outside, but we don't have it looking like that, so let's keep going. All right, we don't care about that. This is Kimberly done. Okay, so what do we have here? Well, we have, what do we need to know? We need to know the ciliary body. So the ciliary body is going to be around here. See that kind of edgy part there? Almost like it's serrated. Be careful. This part here is the iris, right? You guys see that difference? And do we need to know the iris? We don't. So ultimately, it doesn't matter because in this particular image, the only thing they're going to ask, the only thing you have to know is the ciliary body. Okay? That's it. Okay? The iris is just going to be this part, but we don't have to know the iris, so you're fine. In this image, we could also, this image is so weird. So the sclera is nicely in view from the side, right? So we can see the sclera. Theoretically, we could ask for the choroid here. See this black part? So technically, there's some choroid over here as well. The black is choroid, but not up here in the front, guys. Up here in the front, we're looking at ciliary body around there. Yes, Gloria?
So would this be choroid right here?
It would be. It would be. Yep. We could ask, but I doubt it. I think we're going to go to another image. So that's why you can see it there, and you can also see it here in the black. And of course, the sclera is this hard, white, kind of smooth part here. So we wouldn't go to the kind of fatty parts on the outside there. All right. And then this image is so strange because the beigeish part is the retina, but you could say that maybe there's some vitreous humor back here. Not pretty vitreous humor because it should look like this. Yeah, it should look like that. It should look like the gel. So theoretically, so to me, this is a lens, and they popped it out of here. The lens was sitting here. They popped it out, and when they popped it out, obviously the back of the lens, there's vitreous humor. So this is what vitreous humor looks like there. So, and then all this very light, and when I mean light, I mean both color and width or thickness. It's very thin tissue, the retina. So that's what we have. But I guess they could ask for the vitreous humor here because they don't ask for the lens for some reason. That is not the cornea, by the way, guys. That's the lens right here. I know that because I popped them out in the past. I'll show you the cornea in a second. All right. So this image is great. Why? I think they're going to ask for the choroid and the retina here. This black part and this iridescent, beautiful blue and green, that's all the choroid. The retina is what they're holding here. Do you see that, guys? So they've taken their, actually these are just scissors. No, they're not. They're actually forceps. So they've taken the forceps, they've taken that retina, and they've started to peel it up. So you can see how thin it is here. Beige, that's the retina. The black and the iridescent is the choroid. Okay, guys? Theoretically, you can still see the kind of serrated portion of the ciliary body, and you can see a really nice sclera here. Look at that. Pretty. I don't know. Well, if we think about light refraction, right? The black part would be something that would have the melanin to absorb light. However, that iridescent would kind of refract light, I would think. Oh, get out. That's right. I forgot. You're right. Melinda, did your teacher tell you this, or do you just know agriculture? Actually, that is correct. Correct. That's right. Thanks to Melinda. Probably. That's awesome. That's right. Because they can see at night. That's right. That iridescence, though, but I think I was right. That iridescence is going to do more refraction. So, the little bit that they can get, they can really have more acuity. Gloria, yes.
Question 999. On the other image, can you show us the retina one more time so I can compare it to this one?
Yeah, sure. So technically, it would be, there's kind of a little bit more of a gelatinous substance, but can you see it in the background there? Can you see a little bit of the beige underneath there?
Okay, I see that. And so the vitreous humor would technically also be there.
Technically, yes. I think they would probably go with the vitreous humor here because it looks like there's something over the retina. However, and I don't know if they would do it here. I think they probably will want the vitreous humor here, even though it should look like this a gelatinous substance. But you can see that retina is not very clearly visible as it is here. So yeah, I'm going to go with high probability of vitreous humor here, possibly here because that's what it is, but probably here. And then, of course, and then they're going to go with retina here. Yep, awesome. All right, good job. And cornea. That's it. Awesome. And this is the one for the optic nerve. And then you can see the cornea here as well. All right, guys. So with that being said, are you ready? Ciliary body. And this is actually nice because you get a nice ciliary body here. Good. Possible sclera. Yes. Probably vitreous humor. Yes. Possibly vitreous humor. Yes. Possibly choroid, but probably not. Yeah. All right, good. That's it. Yeah. Oh, careful, not the black. Yeah, it's got to. I just don't think they'll do it there. I just don't think so. All right, good, but that's good. All right, definite retina. Yeah, definite retina, and then choroid. Yeah, they'll either do the iridescent or the black part, and probable sclera actually, because they peel back that sclera a bit. It's really pretty there. Good. Possible ciliary body. Yeah, you can see it there. Although I think it's in the other one. All right, and let's go. Cornea. Yep, that's it. And optic nerve. Possible cornea. Yeah, unlikely, very unlikely. All right, did we do everything? We did. Yes, good job, guys. Awesome. All right, with that, we are almost done. The eye. The microscope slide of the eye. Okay, so let's go ahead and knock it out. Oh, question?
Miss Nicolette,
Yes.
Sorry.
No,
Can you go back 1 second and point out that nerve again? Please?
Yes,
Thank you.
absolutely. It's your optic nerve. Do you see that? It's actually, so they have to, okay, this is going to be gross, I'm sorry. But when they take the eye and pull it out, they have to, they're going to they're going to pull out or cut the optic nerve because obviously it's attached to the brain still. So it just looks like a nub. And it's hard when you feel it in lab. So that's it.
Okay.
Pupillary reflex. Yes, thank you for asking. We haven't got to any of the reflexes yet, but basically the reflexes, okay, so let's deal with the eye experiments now before we get to the next one. So the eye experiments, they're all in the lab manual. So if you guys go to page 155 and 156 is eye physiology. And we don't actually do these, we don't do them over Zoom. And there's really not much to do in the lab. We can do a little bit of them. So what do you need to do for those? You need to read them from the lab manual and just know them. That's it. This is like a lecture note thing. Okay? So in other words, just read the acuity, understand what it means, and be able to answer a short question about any of these. That's it. Does that make sense, guys? So for example, for the pupillary reflex, it's repeat the pupillary, wait. Hold on, come back. Where's the pupillary reflex? It's not like repeat the pupillary reflex. That doesn't make sense. Where is this? Hold on. Oh, okay. Page 148. It says it right there. All right, so on page 148 is the one I chose. It's not quite there. So on page 148, there's a pupillary reflex. So, and the pupillary reflex is basically just put a light to your eye. Whatever happens on the right eye happens on the left eye. Okay? And you're going to get either you're only going to get one thing. When you put the light into your eye, the pupil is going to constrict because the eye the retina needs to be protected from too much light. So you close up that muscle, that pupillae, that constrictor muscle, that constrictor pupillae muscle, in order to close the diameter of the pupil. So that was just an overview. But literally, simply read all of those reflexes. If you need to take little flashcard notes and remember the key points, do that. We're going to ask about one of them, maybe two. But read it in the lab manual, and that's it. Estella, does that work? Literally, just read it, study that, boom, you're good to go. Yeah, I know. It's kind of, it is what it is, to be honest with you. It just is what it is. But it's good to you review that. And it's going to be something that you can be asked on the lecture, so knock that out. All right, so now we're going to go to the microscope slide. All right, so this is kind of reviewing what we just did. Remember when we went at that image of those 3-cell layers of the retina for the flat eye model? Well, these are those layers. They go like this: A, B, C, pigmented epithelium. Okay, so same idea here. And again, A, B, C, with A being the ganglion cell, B being the bipolar, and then C being the rods and cones, and then we have that pigmented epithelial layer. Okay, a single layer of epithelial cells. Now, here, they want it a little bit different on the slide. What they want is they want rods and cones, and then they want nuclei of the rods and cones, and then they want nuclei of the bipolar cells, and then they want nuclei of the ganglion cells. Okay, that's fine. We're going to see what that is. Okay, and so let's go ahead and kind of knock it out here. Okay, you know which direction we know. So I've already done this, just FYI. Here's the A, which is ganglion, B, which is the bipolar, and then C, which is the rods and cones, and here's the pigmented epithelium. Okay, we did that already. All right, I've got a couple slides. Now, this is from Soft Chalk, so FYI. And this is the 3 layers, because they do ask for the sclera, the choroid, and the retina. It's no problem. Why? The retina is going to have these 3 layers of cells. 1, A, 2, B, 3, just a couple peeking out here, C. This A is going to be the nuclei of the rods and cones. B is going to be the nuclei of the bipolar cell. C is going to be the nuclei of the ganglion cell. And this dark part, pigmented epithelium. Okay, guys? That's what we're looking at here. Okay? This is all the choroid. This is all the sclera. This is the retina. Okay, and we're going to see a couple more images of this, and we'll look at that. So here is an image, a darker image, blackboard, showing the 3 layers again. Here's A. Here's A, rods and cone. B, nuclei, and this whole dark dotted layer. B, nuclei of bipolar cells. C, nuclei. Oh, wait. Hold on. Which way did I go? Oh, I switched my ABC again. Sorry, guys. Let me go the way I did it before. I want to stick with the way I did it. Let me do it the way I did before. A, it's right. The layers are right, but I want to keep the same ABC layer. A is ganglion. B is bipolar. C is rods and cones. Yeah, because that's how I did it here. A, ganglion. B, bipolar. C, rods and cones. So I didn't say they're wrong. I just flipped the ABC, so I don't want to do that. I want to stay consistent. So again, let's stay consistent with A, ganglion, B, bipolar, C, rods and cones, with light coming in this direction. Okay, guys? So the layer's still the same. It's just I rearranged the ABC so it's in the same. Okay? All right. I know. Yeah, I just realized that. Thank you. All right, awesome. Now, to say the same with what we've done, A, bipolar, B, ganglion, C, rods and cones. That's correct, but I'm bumping that up. And actually, this image doesn't even matter. So technically, it's actually this one here. A, B, and then C. But don't worry about this image because you're not going to see this image, so I don't want to confuse you. I want to move on quickly. Sorry, that image you're not going to see. This image is what you saw in soft chalk. It's the same one, only all you're seeing is the retina and a peekaboo choroid. Okay, so as our labeling, A, B, C. Okay, now this is different. ABC is the nuclei because that's what you can see the dots. This part, the pink part by the pigmented epithelium, that's what they're calling rods and cones. Technically, it's the rods and cone axons. Okay, guys. So look at this, and this is on soft chalk. So A, same thing here. Same thing. C, same thing. The difference is the part that's not a nuclei is called the rods and cones. It's the axons. Okay, guys, and then the pigmented epithelium, which you see here. Okay, the ABC now is in the order I did it with the flat model. So then it's the same. All right. Don't worry about this. Now, this image looks like the one on Blackboard. I'm sorry, it's not. It's actually inverted. So the sclera is on. So the sclera is actually on this side on Blackboard, but it's the same. It's the same. The image looks the same. So you have the sclera over here. You have the choroid over here. And then all of this is the retina. That's the retina. And this one is labeled. Okay? So, based on ABC, A, right? These are the neurons, the ganglion neurons. B, these are the bipolar neurons. C, these are the rod and cone neurons. And then this part here is what we just call rod and cones, because that's the axons. Okay, and this is nice because it's labeled correctly. Cone nuclei, bipolar cells, technically nuclei, ganglion cells, nuclei. And then, of course, we've got our 3 layers, which are sclera. The pigmented epithelium is all here. And from the pigmented epithelium through here is the retina. All right, guys. Does that make sense? And this one is all correctly. The only thing you want to add for our class is nuclei and nuclei. Okay, so circle the, guys, let's start from there. All right, circle the retina. So that's the retina. So with that being said, where are the ganglion cells? Yes. Those are the ganglion cell neurons. Okay, you're good. Where are the bipolar neurons? Now, there's a lot more neurons, nuclei, in the bipolar layer. That's it. Okay, that's the bipolar cell nuclei. Now, where are the rod and cone nuclei? There's a lot more rod and cone nuclei. Exactly. Now, where is the just rod and cone, but actually it's the axons? That's it. Now, where is the pigmented layer, the pigmented epithelium? That's it. That's it. Where is the choroid? That's it. Where's the sclera? That's it. Boom. The reason why, do I have that? You know what? Hold on. Let me see if I can share it. Hold on. I can get it. Hold on, guys. Give me 1 second. Wait, did I lose you? Hello. Can you guys hear me? Oh, good. I didn't lose you. Okay. Okay. Give me 1 second, guys. Take a look at that. Here, I'm gonna give you something that's a lot less dense. Look at this. Okay, this is from Blackboard. It's from Soft Chalk. Hold on. Okay, hold on, guys. Hold on. Okay, give me a second, guys. Okay, got it. Here we go. And this is just as pretty. Whoa, there's so many more nuclei. Okay, hold on. Okay, how am I going to do this? I can share my application with you, which is Chrome tab I. All right, guys, can you see that? Hold on. Can you see that? Okay, awesome. Now, can you see my pointer? Yes or no? No, okay. Okay, hold on. Oh, I don't want to leave. Okay. Oh, what? Oh, I know. I got it. Okay, can you see my pointer now? Hold on. Yes, okay. So I won't be able to see your questions when you ask them because I've got to go to the other screen and point, but I'll come back. So hold on. I'm still here. All right, guys, are you ready? Okay. These cells here. So first of all, let's look at the whole thing. Sclera, right, guys? Tough connective tissue. That looks like sclera. Choroid, okay? Pigmented epithelium. So all of this is retina. So we're going to go back here in the A, B, C direction that we did with the flat model. Ready? A, all of these are the ganglion cell nuclei. And we're going to go to the next layer. B, all of these are the bipolar cell nuclei. Then we're going to go to the next layer. All of these are the cone and rod nuclei. And then here, these little ones here, that's the cone and rod. So the image I showed you that's labeled is simply flipped the other way, which I guess probably can be confusing, but it's the same thing. All right. So
say it again, please.
No problem. I wish I could label it, but I don't see where I can label it because I've shared a Google Chrome with you. So this one here is A, which is the ganglion cell nuclei. Okay. This one here is B. This is bipolar cell nuclei. This one here is C. This is rod and cone nuclei. And then these pink lines here, just rod and cones. And then the dark line is the pigmented epithelium. The dark line is the pigmented epithelium. Let me show you again. Right
That's
there.
perfect. Thank you.
Was good? You're welcome. I'm glad. Yeah, so the other image, I hope it's helpful now. It's this one though is, yeah, it's flipped. So the retina is, sorry, you can't see it. The retina is here on this image with the pigmented epithelium, where the choroid's here and the sclera is here. All right. Does that make sense? Is that better? Less headachy?
It was great.
Oh, thank you. Sclera is pink. Now, keep in mind, guys, this is really lovely connective tissue. This looks like connective tissue here. It looks a little bit more dense regular to me. Connective tissue. And then the darker part, kind of like Neapolitan ice cream, sorry, but that's what I see, is the choroid here. I don't know. Okay, so light does not go beyond the pigmented epithelium. Light travels past that. You have it mostly right. Light travels past the first layer of cells called the ganglion cells. These are nuclei. The ganglion cells do not absorb light. They also do not absorb light. And they keep going to this third layer of the retina, which is called the photoreceptors, which I'm showing you now, or called the cones and rods. These are the nuclei of the rods and cones. They absorb light. And then that undergoes, this area here is the rods and cones are going to depolarize, hit threshold, and then they're going to send a signal up here, and then they're going to cause a depolarization because of the neurotransmitter on the bipolar. Then the bipolar cell depolarizes or synapse and cause the ganglion cells to depolarize, hit threshold, cause an action potential. All of these axons from the ganglion to the optic nerve, and they head back to the brain. Awesome. It's a good question.
One more question.
No.
I'm sorry.
I was kidding.
So on the exam for the final, what magnification will this one be at? Because I think what I have in my PowerPoint that I've moved over, I don't think I have it magnified at the right one. And I just want to make sure when I'm studying it, I'm not like pulling my eyes. They already like dead right now.
That's actually an excellent. So basically this is what you want to do. You want to go, just to make sure you have the right one, go back to lessons. Go back to the one that says like all these images for the exam and images. And then you're going to go to the retina histology and it's going to come up. Don't touch it. It's going to come up at exactly the right magnification. It looks like it's 20 times magnified. So I would leave it exactly where it is, or the way you see it now. Does that make sense? So don't click around. Just as you go in. Perfect. And then actually,
clicking around.
Don't click around. Just leave it there. And actually, you're gonna. Can you see my pointer? I don't know where I am in this. Can you see my pointer here? Now, do you see what just came up there? Click on that. I will download the image exactly the way it looks.
Okay. Got it.
Yeah,
okay.
So I would not click around. Yeah, good question. That's a good question. I mean, it just is. That was it. I just checked. So when you click on it, just let it go. It'll come up. Don't mess with it, and then just download it. Gloria, go for it.
Yeah, that's the same thing that happened to me. That's why I was so confused when I was looking at the image because it wasn't magnified.
It didn't magnify for you when you clicked on it?
I haven't tried it yet.
Wait.
I just zoomed in on my iPad and like try to write it in there, and then I'm gonna go back and fix it.
Yeah, all you have to do is like when you go back and like click through everything. These are the images. Click on histology. Go to retina histology, and it's gonna come up. Don't touch. It's gonna come to this magnification. And at least from my, I would say they're going to test on this image because it's big enough. Good, perfect. No, I think that might have been the issue, that you got that, you're looking at a smaller image, which is really, really hard. All right, so if everyone's in the right image, that's great. You just click on it, give it a second to come up, and then, like I said, I'll go back. And then you guys can just click on this image here. It'll download it. It'll save it. You don't have to paste. It'll save it at that magnification.
Where's the retina on this one again?
The what?
The retina. Has it retina under microscope of the eye?
So watch what I'm going to do here. The retina is from here to the pigmented epithelium.
Okay. And then the white is the choroid. No,
and then this part is the choroid.
The
You guys see my, yep. And then this one here is the sclera. Now, all of this white part is going to be vitreous humor. Think of it that way. The vitreous humor kind of thing. Does that make sense? So remember this, guys. Kind of just blank space / vitreous humor, like up here. And then we go right into the retina here to the pigmented epithelium. Then we do choroid. Then we do sclera. Can you guys see my pointer? Sclera? Yes, hold on, I gotta check again. All right, good. All right, does that make sense, guys? Sorry, I can't write on this, but you're in a Google browser, so I don't know. Maybe I can, I'm just not that savvy. Yep. Good. And I think that might have been the problem as well, orientation. So yeah, are we good, guys? So we're talking about this whole thing here is the retina, pigmented epithelium. This whole thing there is a choroid. This whole thing here is the sclera. This whole thing here, think of it as a vitreous humor. Good. Oh, happy.
Now, I just followed your steps, and it came up just exactly the way it shows on your screen. So yeah, I think a lot of people are studying it at a different magnification.
Ooh, okay, that's good to know. So this is good. That's why you guys come to class. So, all right, awesome. Now, just in case it's full, I'm going to stop sharing that screen and show you what I had, which is just inverted. Now, I don't want it to be confusing, but it's inverted. So instead of having the sclera on the right, the sclera is on the left, which again might be confusing to some people. So I don't know. Just ignore it if you don't like it. Hold on. Okay. But it's a good point. So forget all of those. Forget all of that. So this one is like your image, only they have a lot more ganglion cells. So on the other image, the sclera is just reversed. So this whole thing, which I can write on now, all of this is retina. And you know, because it's got all dots, these 3 layers of dots, right? Those 3 layers of dots. And then it's inverted. This one is the choroid and then the sclera. Your different section. But if you don't like this, just ignore it. Don't worry about it. All right. So that's it. It's a good point, and it's good. Sometimes we have to kind of dig into it. Like, why am I not seeing the same thing? Why is it so difficult? Blah, blah, blah. So it's good to bring that up. All right. I might do this again on Tuesday with my other class because I think they were having similar problems, but I don't think I understood their problems, to be honest. Yeah, I think they were. All right, so this is the ear. So guys, we knocked out the eye model, flat eye, microscope slide. Remember the eye experiments, guys? Just go back into the lab manual. You know, that's okay. Just know the idea of it so that you can answer a basic question on it. Okay. All right, so we got the ear. The ear, just so that you got kind of some orientation around this. So the ear has 3 parts. It has an outer part, which you can see here, which is basically the fleshy elastic cartilage part, which we'll talk about the name in a second, the pinna. And then here we've got the canal, and this is actually where the sound wave goes in. And then here we've got the tympanic membrane, which pretty much separates this external. Now, this tells it the outer ear, but we want you to know, because it's the lab manual, we want you to know that's called the external ear. And then we've got the middle ear, which is pretty much, it's more between the tympanic membrane and then the area of the cochlea and then the vestibule. Okay? So here you can see they kind of extended a little bit, but generally we're thinking about the middle being more where these bones are, which we'll see more in just a minute. And then we've got the inner ear, and that's where the hearing apparatus, the cochlea, and then the balance apparatus, the vestibule and the semicircular canals are. So that's basically what we have. So let's just kind of knock this out. This is much simpler than the eye. I don't know why. Maybe because it's kind of more linear as opposed to kind of like a ball, less of a sphere, and so it's easier to grasp. But it is what it is. Now, we are not yet into Kimberly Dutton, so just kind of giving some labels to it, so you can take a picture if you like it. So here is the ear itself that you can see, and that's obviously part of the external ear, and that's the pinna. And then we have the external auditory meatus, but they prefer the term external auditory canal. It's the same thing, right? It's like we saw the meatus when we looked at the bone. So the external auditory meatus then is actually the canal in your lab manual. So always follow the lab manual. Things are slightly different depending on the publisher or whatnot. So, and then we've got the tympanic membrane, which we'll see in more detail. And then I'm going to show you the inner part in a more detailed manner in a second. But here's the auditory tube. So, guys, my first little heads up. Be careful between this tube, which goes from, there's a hole here in your ear, of course, and it goes through, and then it meets the tympanic membrane. Okay, that is called the canal, the external auditory canal. Here, auditory tube, and we call it, is it the same thing? And then here, it's the same thing, but it's also called the eustachian tube. So this auditory / eustachian tube, picture whichever name you like better because it's in parentheses. This one does not have to do with sound coming in. We're not going to ask you about that, but I just want you to know that there's a difference. We're going to talk about it in lecture. This comes in via the auditory canal. This is for pressure, exactly, so that you can depressurize your inner, the middle ear area so that you don't burst, basically so you don't burst your tympanic membrane. As you all know, if you've ever been on a plane, as you go higher in altitude, the pressure increases, and that is pressure. Some people, that's why people like chew gum, they suck things, different things, they kind of move their jaw a little bit, but ultimately that's the physiological purpose of the auditory tube or eustachian tube, okay? That's going to depressurize that area so you don't build up in pressure and then cause damage, physical damage. And that's what we have there, okay? So, Kimberly Dunn. So, of course, here we can get the pinna, external auditory canal, the auditory or eustachian tube, and the tympanic membrane. Now, we do have the malleus, which is the first of three small bones, the malleus, the incus, and the stapes. But I don't know that we would ask it here. I'm going to hold off on that. Oh, no pointer, sorry. Thank you. All right, so thanks for the reminder. I see a pointer, so I'm like, you can see a pointer, but you can't. So here is the pinna, which is the outside ear that you see, right? Basically, kind of, it cups in like that, so the sound, just like the cornea brings in the light, the pinna brings in the sound, because the sound waves go everywhere, right? They're in waves. And so we want to kind of bring in those waves into the area called the external auditory canal, okay? And so then with the tympanic membrane, again, this is the malleus, but I just don't think they're going to ask it, so just keep that there. But this is definitely, they can ask these three, well, four. And then the auditory tube. All right, guys. Pinna. Or I guess it could be pinna, but whatever. We tend to go up kind of on the flappy side. All right. External auditory canal. And that's the name we want, even if you see a label differently. All right. Tympanic membrane. Good. I don't know that it's good here, but it's fair game. And then the auditory tube, which is definitely fair game here. Perfect. That's basically it. Okay, great. Now, here, I don't understand this picture because really, what can we ask? The pinna, that's it. Yeah, that's it. So, hoorah. All right, now, yeah, is there an ear here? Okay, there's no ear here. You can't. don't. You're gonna laugh, but I don't want you lose points. Someone's gonna put ear. Oh Lord. Oh no. I mean, I don't even think we'd ask for external ear here. I feel like that would be weird to ask for the external ear here. Oh yeah, guys, sorry, I forgot. We could ask for the external ear here. Right? Yeah, so silly. So yes, I almost forgot. We could ask for the external. And actually, now that so that was funny, but actually good, because theoretically, I think we could ask for the middle ear here too, guys. I think we could. Because they've excluded the inner ear here. So guys, don't be confused. Right? I don't think it's confusing, but this is external. Right? All of this is external. This here is middle, not internal. We got to go one more layer, which we'll see in a second. You can't see the internal layer here. So that was actually really good because I forgot that. Yeah, but that's what it is. External, middle, and internal ear. All right, booyah. We don't care about that. All right, so here is just a kind of explanatory illustration. Why? Because here is the middle ear, right? So you've got the tympanic membrane here, which we saw, and then you've got this part which begins the area of balance and hearing. Okay, so that's the internal ear, but the part the middle ear. Okay, now what's in this area? I was going to say what's in here, but then somebody was going to say something. So what's in this area? Well, we've got those three bones that you're going to learn, and you probably know them. Together, they're known as ossicles. They're very thin, very fragile bones. Yes, hearing apparatus. And they're called the incus and stapes. So they're in that, you can think of it like this, right? So what do I mean by in that order? Well, the malleus, which is supposed to look like a hammer, I think, like a mallet. I don't know, whatever, you be the judge. So it's the one that kind of has a fatter head here, and it attaches to the tympanic membrane. Okay, so that is an attachment there. It's very small, but it's the first part. So anything attached to the tympanic membrane, that first bone is called the malleus. The second bone, which is a very small one, but that attaches to both. So the second bone is the incus, and it attaches to both the malleus and the stapes. So that is kind of it's the middle bone of the three. And that's it. The third bone of these three, going from the tympanic membrane into the hearing and the balance, is going to be the stapes. Now the stapes is easy because it looks like a door handle. Basically kind of looks like that. So that's great. Or a stirrup, if like the horse's stirrup, sideways, whatever. Because it attaches a bed staple. Awesome. I like it. It attaches to something called the oval window. Okay? So technically it's more like this, and then it attaches to this thing, which is called the oval window, which you image. So oh really? I don't see that.
When you said stirrup.
Oh!
Repeating.
Oh! I forgot about that. I think I need to go see my doctor. Anyway, it's been a hot minute. So I was like, taking care of. I don't even ride horses, so I don't even know where that's coming from. Anyway, it's called a stirrup. And yes, at the gynecologist, there are also stirrups that you put your feet up. I was like, wait. All right. And then, so that's what we have here. And we're going to talk about the oval window compared to the round window in just a little bit. Just know that in this middle part of the ear, we're going to have the first bone called the malleus, right? Kind of like the head, the hammer, attached to the tympanic membrane. The next bone, the incus, is attached to both the malleus and then the stapes. The stapes looks like a little, whatever you think it looks like. And that is attached to the oval window. And then the round window is separate. It's just close to it. Okay, and then there's the auditory tube. And so here is where you are in the ear. Okay, with that introduction, let's go ahead and look at a couple more. I like my pictures. So this is a nice one. That looks like an actual image. Maybe maybe an X-ray, possibly. That looks like definitely some image of the Malleus, Incus, and Stapes. And you can see that what we kind of see mostly here is this part. The Malleus is attached to the Incus. I should probably choose another color. The Malleus is attached to the Incus, is attached to the Stapes, and there's the Stapes attached to the oval window. All right, so I like this image actually because you never see them separate. So depending on the image, it's kind of hard to look like. All right, with that being said, you can't see all of them. The only thing you can see in this image is the Malleus. Okay, so Incus and Stapes are kind of tucked behind it. But you can see this, which is the external auditory canal, not a tube. It's not an external auditory tube, right? Because the auditory tube is, where's the auditory tube? That's right. So just be careful between that canal, external auditory canal, and the auditory tube. Good. Now, in image, yes, exactly. In this image, you can see a few things which are new as well. So we've got these things that look like roller coasters, and there's 3 of them, and they're called semicircular canals. Now, it's kind of nice, right? Because the semi means part circle. So here you've got part circles, semicircular canal, okay? And then they've got these things called ampules, which we don't actually care about in lab because we're not asking to label it, right? But that's what they are. So we've got the semicircular canals. That's great. And then we've got the vestibule, which you can't really see here. It's actually kind of in this area here. So the semicircular canals sit on the vestibule, okay? And it's not a super view of the vestibule, but you do have obviously a good view of the tympanic membrane, of the external auditory canal, of the auditory tube, and yes, the Malleus, okay? And the 2 other things you can see, but not super, is here's what looks like a snail shell, and that's the cochlea. That's the hearing apparatus. So this is where we hear, and this is where we do balance, right? Vestibule and the semicircular canal do 2 different types of balance. One is static, one is dynamic, which we'll learn in lecture, so don't worry about it here. But just know that this area is balance, this area is hearing, okay? And this, which you can see just one of the merging, this is actually the cochlear nerve, but together they make the vestibulocochlear nerve. Okay, so that's kind of legitimate. All right, guys, where are the semicircular canals? Awesome, that's it, all there. I'm hesitant to say this the vestibule, but let me show the vestibule. Where do you think the vestibule are? Yeah, so it's down there, but I don't love it. It's not the best image. Where's the cochlea? Good. Vestibulocochlear nerve, because we don't separate them. They're just one nerve because they merge. The vestibular nerve and the cochlear nerve merge. All right, and then just again, auditory tube. Yeah, oh, not the canal, the tube. Yeah, that's the tube. All right, so this is not good. This one is not it. That's the canal. Good. This is the auditory tube. All right, awesome, guys. Now, just in case, so we're going to see an image in this orientation. So this is actually a nice image because, you know I like my labeled images. Here you can see the Malleus, the Incus, and the Stapes. It's really pretty. And the Stapes is attached to the oval window. Here you see the very pretty semicircular canals, and you see a very clear vestibule. Here you see a very pretty cochlea, and here you see a very nice, together they're all the vestibulocochlear nerve, the whole thing, okay? And then, of course, the tympanic membrane, and we could ask about the auditory tube. All right, guys, so if you want, take a picture. All right, ready, similar orientation. Where are the semicircular canals? So I think they're going to go vestibule here. Why? Because it's very clear. So where is the vestibule? Technically at 43, but don't memorize the number. Yeah, so it's right here. Oh, here. That's the vestibule. Good. Now, where's the cochlea? Snail shell for hearing. Good. Now, where is the vestibulocochlear nerve? I literally have to say it like that. All right. You can see the tympanic membrane. Where would that be? Yes. You can see the Malleus and the Incus, but I don't love it. So the first part is the Malleus. The second part is the Incus. So Malleus is the little, that's the Malleus. And then the second part is the Incus. Yeah, that's the Incus is right over here. Okay, awesome. All right, but I'm thinking they're going to go more inner. Now they could ask for the inner here. They could ask for the inner ear in this area because that's the inner ear. Just FYI. All right, good. And then here, this is also labeled. It gives you a nice. Actually, this is very nice. This is quite nice, similar to what you saw. So all of them all labeled here. You see, they did a good job with the stapes, which is straightforward, but also with the oval window. So that's good. Watch your oval, guys. Watch your arrow, right? If they're going in close, it's an oval window. If they're coming out like this, it's a stapes. Okay. All right. And then they show a nice malleus, incus. All right, guys? This is a good image. All right. So here we go. Yes, here they can ask for the malleus and the incus. So where's the malleus? And I keep saying incus, but it's incus. Good. That's it. Where's the incus? That's it, right? Now you can't really see the stapes here. You just can't do it. But where's the tympanic membrane? I don't know. They could ask. Yep, the pink and the kind of it's mostly it's the pink, but also it's the clear part in the middle. All right, semicircular canals. Oh, go up. Roller coaster, roller coaster, roller coaster. Awesome. Vestibule. Vestibule. Yeah, there we go. I like it. Cochlea. Yep, that's it. Vestibulocochlear nerve. All right, circle the inner ear. Yeah, careful, not too far, not too far. Circle the middle ear. Whoa, middle ear. That's it. Don't go too far. That's it. Awesome. Boom. I like it. All right. Good job. I don't know what I'm doing here. Okay. Good job. Very good. Now, super easy. Where's the malleus? Yep. Careful. It looks like a big bulbous head. That's it. Incus. Good. Tympanic membrane. They're going to point to the clear part. All right. Awesome. Good job. Now, this is just a big, I don't think you need this one. It's all labeled, but hoorah, let's go. All right, so I have this so that when we get to the round window, it makes sense. So obviously these are semicircular canals. Now, here is where we'd have the stapes, right, guys? So what you see here is just the oval window. So oval window is at the stapes, right? And then here, you can see the round window. That's why I got that, because you can see the relationship to it. All right, so let's find this sucker. All right, find this. Super easy. Oh, careful. Come back. There we go. Awesome. Now, with that being said, where's the oval window? It's kind of like a beige. Oh, don't go too far. You're going too far, guys. That's it. Just there. Watch, guys. Watch me for a second. If I can get some color. Stapes. Okay. Oval window. We good? Tell me where the oval window is. That's it. Just right there. Sweet. Now, are you ready? Are you ready? Round window. You got it? Stapes, oval window. And then round window. All right. Where's the round window? Not oval. Where's the round window? Round. Go down. Not at the stapes. Not the stapes. Oh, careful. So the stapes looks like this, guys. So it looks like it's got a hole in it. That's not the round window. So watch what I'm doing here, guys. Watch. Stapes with a hole in it. Dude, it looks like a stirrup, remember? And then oval window. Boom. Yeah. And then other orange part, or whatever color you see, is going to be the round window. You got me? Do it again. Stapes. Good. Oval window. Yep. Stay with the stapes. Good. And then round window. Down. There we go. That's it. Boom. All right. With that being said, we can't really see the oval window here. So where is the, sorry, I lied. We can't see the round window here. Where is the oval window? Boom, that's it. Boom, 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 boom. That's right, because this is what? What bone is this? It's either the malleus, incus, or stapes. It's the stapes. Boom. All right, but we can't see the round window here. We can see it. Let's go back. We can see it here. That's the round window. Okay, guys? Did we? Oh, frick, I hate the antrum. We didn't. Okay, so the antrum's janky as crap. The antrum is basically an area. It's like it's a space. So it's basically a space in the middle ear by the stapes. It's just a space. I mean, I have looked everywhere, and where the hell is the antrum? Like, I searched everywhere. Literally, when I first, searching with these models, I was like, where's the antrum? It's like a space, and the best that I could, it's like close to the stapes. And I'm like, are you freaking kidding me? I don't know that I've ever seen them ask it, so I hope they don't. Yeah, it was freaking, it's crazy. I have an image at the end of this slide deck, which I won't even show you, because it's stupid. So what would they use for that? Let me see what they would use. It would have to be an intact, so that's my image, which is freaking crazy, because it doesn't even make sense itself. So I think they would use, so potentially they could use something like this, and they could like point between that area. That's the only place they could really do it. The everything I said that I read, it was in the area of where this close to the stapes. So again, I've never seen it on a test, but just that's where it is. So yeah, sorry guys, that's what I got. All right, so that's it, antrum. Yeah, and I never talked about it for that reason. Okay, other than that, that's it. Okay, so are you guys ready? Let's knock out the muscle. There. How what? For the antrum? Oh, sorry. Remember, sorry. So I wanted to go over the muscle fiber because we really didn't go over the whole thing. It looks like this, guys. Hold on. Oh, do you want to call it a day and do it next week? No, you want to just do it now?
Yes, let's do it now. I need to study.
Okay. All right. Okay. It's not going to be that bad because we hashed it out last Tuesday. It was a pain in the butt. Okay. So, okay, guys, let's do it. First of all, forget everything. I'm just going to go right to our model. Okay. So let's do a couple of these. These are all our model, Kimberly Dunn. Okay. So the easy ones. Oh, sorry. I was about to do that. Yeah. Sorry. 80. Hold on. 82. So striated muscle fiber model. That's this one. Okay, guys. This is all one muscle fiber. Okay, that's one cell. Have inside it myofibrils. Not a good color. Myofibrils. Right? Boom, 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 boom. Done. Oh, no, that's okay. Let me see. Hold on. Oh, yeah, it's not this one. Oh, wait, is it? It's the same one. Isn't it the same one? I actually think it's the same one. Yeah, it's the same one. No problem. You're right. It's just I saw it over here, but no problem. All right, so 2 or 101, no problem. We should be the same. Okay, so easy ones. Myofibril, no problem. Boom, we got it, right? So we're going to knock out, okay? So I'm going to jump back and forth. Okay, actually, I'm going to come back to this model. Don't worry about it. I'm going to do all the easy stuff first. Okay, here. T-tubules. Boom, 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 boom. Because the top of this sarcolemma makes the T top, right? That's it, okay? Easy. Sarcoplasmic reticulum. It looks like the net over here. Boom, 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 boom. Easy, Okay, no problem. Easy. Mitochondria. Okay, guys, watch. This is the sarcolemma. This is the sarcolemma here, which means this is the cell, the muscle fiber. These are mitochondria. Okay, this is the nuclei. Bugs in between. Yeah. Sure. Let me see if I've got a bigger model. Hold on, guys. Here. No, that's not ours. It is the same model, but it's not ours. Hold on. Okay. Okay. So what I just said was sarcolemma. In other words, the cell membrane of the muscle fiber. You got me, guys? See that? Mitochondria. They always